One of the things that I would say, if a CRO takes more than 48 hours to get an NDA signed, it's a wrong CRO. We're going to have problem. If they say we're going to have 6 months before we get your project started, if they'll take 2 weeks to give you a proposal, one of the things I'm very proud of, we turn around NDAs in 48 hours, often less, and we promise to develop a proposal within 48 hours to 72 hours.
Welcome back. In part 1, Ron Najafi walked us through a career built on scientific rigor and entrepreneurial conviction. Now we go deeper into the practical questions that matter for your work. How to structure a productive CRO partnership, what separates a boutique specialist from a full-service organization, and what the future of pharmaceutical analytics actually looks like. Ron also shares the hard-won lessons that 3 decades of building companies tend to produce. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brolman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Besides the impurities, now as you're working with various clients, what other challenges do you see in the analytical field where you help your clients as a CRO?
Some of the things that we've gotten involved is doing a pharmacokinetic, for example, for a small molecule is relatively straightforward. But if you're doing pharmacokinetics in the eye, in the vitreous, which is the liquid inside the eye, which is roughly about 1 or 2 milliliter, you need to develop a method. You need to be able to validate that method, and you need access to the vitreous. So we've actually been involved in that kind of activities where we bought vitreous humor from an eye bank, and we got involved in doing that. We actually published that work with a company called Rayner Surgical. If you actually search Rayner Surgical, Emery Pharma, we have significant expertise in ophthalmological pharmacokinetics. And that goes back to my experience with NovoBay as well, where we were involved with ophthalmology, and I have a lot of ophthalmological key opinion leading ophthalmologists near me and friends of mine. We've been getting calls from them wanting to work with us on this project or that project. As a result, we have a lot of experience there. Another one that's I think is significant, is challenging, is to assess a protein. Let's say a gene therapy company is trying to silence a gene, and therefore they're trying to see if certain protein level goes down, goes up. In a very systematic fashion in an animal study, we've actually done that. And these are not your average pharmacokinetics, David. You have to take the blood sample, get to the plasma, basically trypsinize everything. Before you do that, you have to know what's the sequence of your protein, do an in silico analysis of what peptides would be generated as a result of trypsin sort of interaction, and then identify several peptides and then use those peptides as surrogate marker for the entire protein. And then you have synthesized the peptide. develop a reference standard of the peptide and then use it for calibration curve, it's much more involved, much more sophisticated than just a small molecule. So biologics have their own challenges, and we have established very extensive, thoughtful workflow, that systematic workflow that would give us what we want. So if there is a small change in the gene therapy, we would be able to see it. Does it work? Does it not work? All of that by doing the pharmacokinetic study.
Yes. You work in different fields. You do impurity testing, you do pharmacokinetics testing, and a lot of other services. I'm just wondering, if smart biotech scientist is looking for a CRO, what are some factors they need to consider to understand which CRO is a good fit for them? Because there are big ones, there are smaller ones, there are more diverse ones. What is the advice you're giving them?
Well, I would say if you want to have a CRO that's big enough to have the resources, but small enough to move quickly. Some of the larger CROs are very bureaucratic. They have a lot of turnovers. They have workflows, but they don't have expertise to figure out if something didn't work in that workflow, why it didn't work. They use technicians for the most part to conduct some of the activities. One of the things that I would say, if a CRO takes more than 48 hours to get an NDA signed, it's a wrong CRO. We're going to have problem. If they say we're going to have 6 months before we get your project started, if they'll take 2 weeks to give you a proposal, one of the things I'm very proud of, we turn around NDAs in 48 hours or less, often less, and we promise to develop a proposal. If we have all the key informations we needed within 48 hours to 72 hours, we have a proposal to the client, and very extensive proposal. One of the things that our clients are very impressed is how extensive our proposals are and how we lay things out. Of course, we're also more sophisticated activities. We do like peptide mapping, and then we validate it. We do GLP type activities, and that could be submitted to the agency if it's like a GLP tox, or if they needed the data. Definitely one of the things that I would always say is, when you develop an analytical method, the method needs to be validated. Even if it's not GLP, you want to validate a method because there's a reason why FDA requires validation. We want to make sure the method is robust. If you get a frozen sample, have you tested the stability of your compound frozen? Have you? I asked the clients, do you know if they have no idea. So you want to check the stability of your product frozen, basically do a freeze-thaw stability, do room temperature stability. All of those things points to a good analytical method.
I would like to ask you about how do you develop a good relationship with your customers? Because when you're choosing a CRO, or the same applies for a CDMO, this is a relationship you establish that will go on sometimes for several years. So it's almost like marriage. So how do you go about that?
I think managing expectation is number one thing. I always tell my team to make sure if we have any challenges, analytical methodology, if we're going to have delays, if an instrument breaks down or needs to be serviced, we try to maintain a close, honest relationship with our clients. We work as if we're part of their operation, part of their team. From the moment the proposal gets signed and goes into our chemistry group or biochemistry group or biomarker group, we have a scientist that they can reach out to, and that scientist updates the client sometime once a week, sometime twice a week. So we have a very close relationship, very honest relationship, and we collaboratively try to solve problems. At the end of the day, we need to develop trust. And that trust comes by us showing our experience, our expertise, and utilizing those experience and expertise and managing expectation. Then you develop the trust.
Yeah, absolutely. But developing a trust is key in any customer relationship or in any entrepreneurial endeavor. I would like to go back to your entrepreneurial story. Just ask you, what were, I'd say, the number one or two main key lessons you have learned as an entrepreneur?
Well, I've had multiple chapters. If you asked me about fundraising from the days that I raised money for Noble Bay, I raised $100 million from private and public investors, and I brought in $100 million in partnership revenue that came through Noble Bay. On that front, I will say, raise more money. When you have an opportunity to raise money, if money is offered to you, don't negotiate too hard on valuation and so forth. Take the money, because having that money will be more important than not having it, because you don't know what's going to happen as you go through clinical studies, as you could have hiccups, you could have challenges. So number one, raise more money than you need. Number two, make sure you bring on board good people who are creative, flexible, hardworking, smart. And that's also in the absence of not knowing where you're going. If you're lost, smart people are the best thing to have. If you're lost in a desert, it's better to be with a bunch of really smart, entrepreneurial, creative, positive-minded people. So those are two things that comes to mind from my experience at Noble Bay. The third thing is, I would say, when you have an asset and you want to partner that asset, make sure you have the right people helping you with mergers and acquisition, with partnering. You need to have right skill set to actually find those partnerships. And it took us a while to get that, but that's really, really important, because that creates a lot of value for you down the road, partnership opportunities. Now, in the next chapter, which is really I don't have any investors at Emery Pharma, goes back to the team. We have to make sure the team has the skill set, has the equipment they need, attitude, the morale, the environment, all of that is really important. I think this is also true in any company, even in my previous chapter. But not having investors is a blessing in this chapter of my life, because I don't have to deal with investors and I don't have to deal with fundraisers, and I can go to bed a lot easier.
That's good to hear.
Without investors.
I imagine you have a lot more freedom and independence.
I tell you, one of the things I would say, if you don't have to take a company public, don't take the company public. Having a public company and having a company, it's like having two different entities. You have to deal with the public company, you have to deal with investors, you have to deal with institutional investors, and then you also have to run the company. It's a lot of work. It's a lot of challenges. I enjoyed it. Would I do it again? I probably won't, but I much prefer the private enterprise where you can focus on the company and its goal and its success.
I'd just like to look ahead a few years and I'm curious about your vision of how you see biologics evolve. According to you, Ron, what are the biggest unmet analytical challenges in biologics?
Let me tell you, one of the things, this came up in a recent meeting we had with a government entity that is trying to develop a product for government. And one of the biggest costs in developing biologic is their bioanalytical testing for this product was several $1 million. And the reason for it is they use old-fashioned ELISA techniques. They have 5 or 6 different biologics, and they need to measure all of them using ELISA. Each ELISA needs to get validated, then each of them needs to get tested. Mass spectrometry cuts through a lot of that. And we think we can reduce the cost of biologic, the analytical portion of biologics, probably by 50%. That's going to be a major breakthrough for biologics development. Now, For personalized medicine, that's still a tall order. And I think we need to still develop some methodologies for testing and qualifying for control of some of those biologics. But for larger products, for bigger products that do get commercialized through the traditional channel, I think bioanalytical testing, the price of that could come down by a lot.
Wow. And this is just because the technology will evolve and we will develop also better workflows.
Absolutely. The technology is here. We've actually been doing it. Emery Pharma, we've shown our various government partners that this can be done. This price can be lowered. So I say that with confidence that we could test a mixture of 6 antibodies in blood in one run by mass spectrometry. One run versus 6 different run, 6 different validations, 6 different this and that. We can do one validation, one run, and get the data much more accurate than traditional ELISA technique.
Well, Ron, we have covered a lot of ground today. What is the most important takeaway you want our listeners to walk away with?
There's a lot of excitement, enthusiasm for biopharma, biotech, biologics out there. Really excited about all the peptides that are being developed, be it GLPs or other types of peptides that are being developed. We have a whole host of clients that are developing various peptides for every indication under the sun. And I think the speed of development can be increased. I still think people need to have a better understanding of how FDA operates. Having those experience and expertise is important, how they should deal with the FDA. But I'm very enthusiastic about the future. And please, your listeners, if they like to reach out to me, they can reach out to me via LinkedIn. My email is ron@emerypharma.com. They have to have my email address in order to be able to successfully connect to me on LinkedIn, ron@emerypharma.com. And of course, we have a YouTube channel, Emery Pharma, where we have a speaker series, very similar to the one that I created back at Rumpel & Rohr. Our speaker series, the best of the best, including yourself, who've been a guest of our speaker series, YouTube. And of course, we're reachable through our website. Oh, one more thing, David. I have my digital twin on our website live. Essentially, it's a digital twin of Ron Najafi, and you can ask any questions under the sun regarding what we do, how we do it from my digital twin. We've spent a lot of time teaching the digital twin everything that's on our website, so they should be able to provide you with a lot of information.
Excellent, Ron. There you have it, smart biotech scientist. Used Ron's digital twin, but not only, also reach out to him and his team. You will find the links in the show notes. And Ron, thank you very much for being on the show today. It has been a huge pleasure.
Thank you, David. Pleasure is all mine, and look forward to working with you.
Nitrosamines, CRO selection, and the future of analytics. Ron covered it with the precision you'd expect from someone who has lived these challenges firsthand. If you work in drug development, there is something here worth sitting with. And if today's episode was useful to you, please leave a review on Apple Podcasts or your platform of choice. Thank you for tuning in today, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.